So in the previous video we saw that the absorption of the water takes place by first two methods was imbibition, next was diffusion. Imbibition was due to surface attraction. Diffusion was due to the movement of the water molecule from a higher concentration to lower concentration when they are direct contact with each other. We now move on to the next method of absorption, and that is osmosis. Now this osmosis is the most important kind of the process of absorption. So let's understand the definition first and then we move forward. So defining osmosis is this way. Osmosis is the movement of water molecules from the region of higher concentration to the region of lower concentration through a semi-permeable membrane. So what is this? It is basically the movement of water from higher concentration. That means you have water, more water at one part and less amount of solute over there. You get more amount of solute in another part and less amount of water over here. So, but this both are separated by a semi-permeable membrane, and if then the water from a higher region goes into the lower region, with this membrane being a part and parcel in between, in that case this movement of water from higher to lower in the presence of the semi-permeable membrane is called as osmosis. Got it? So let's understand once again the definition. Osmosis is the movement of water. Okay, osmosis is the movement of water molecules from the region of higher concentration, that is the soil if you consider it this way, region of higher concentration, that is the dilute solution, to the region of lower concentration, that is the cell sap, lower concentration, that is concentrated solution, through a semi-permeable membrane, that is nothing but the cell membrane. So when it is passing from the cell membrane from the soil into the cell sap, that is nothing but osmosis. So osmosis is the movement of water from higher concentration to lower concentration in the presence of a semi-permeable membrane or passing through a semi-permeable membrane. Now what is most important to note in the above definition is that only the water molecules move from a higher concentration to their lower concentration. That is higher concentration means it is a dilute solution, that means the solute content is less and water content is more. To a lower concentration means concentrated solution means solute is more and water is less. So that movement is there in this case. So in case of osmosis, the most important thing is that we see that only water molecules are going to move from higher level to lower level. So that was the important part. Now the water, when it is entering into the cell, so there will be an inward entry of the water. So the inward entry of the water into the cell is called as end osmosis. So inward is end osmosis. And when the cell is going to be having more water and the next cell which is next to it is having less water, so the water from the one cell goes to the another cell, then the water is going out of the cell, that is called as outward kind of osmosis, is called as exosmosis. Endo, enter, exo, exit. Endo, water coming inside, so endo, that is end osmosis, entering of the water inside the cell. Exosmosis, exit of the water from the cell. So inward and outward osmosis, with reference to the cell, the osmosis can be either inward or outward depending on the extent of concentration of the solution surrounding it. So depending upon what is the condition around it, like suppose I take a sugar solution, okay, and I put some grapes inside, what will happen? the grapes will have more water than the sugar solution. So the water from the grapes is going to go out into the sugar solution. So that's why water from the cell of the grapes are going out. So it is outward exosmosis. And if I take kismis, that is dry grapes, and put it inside water, what will happen? The water will enter into that kismis, it will enter into the raisins. So that raisins are going to swell up. So that is endosmosis. Water entering into the cell, endosmosis. So we define them as endosmosis, inward osmosis, is the inward diffusion of water through a semi-permeable membrane when the surrounding solution is less concentrated. This tends to swelling up of the cell. So when endosmosis takes place, the cell is going to swell up because the water has entered into the cell, so the cell swells up. So swelling up of the cell is taking place in endosmosis because the concentration of the water outside the cell is greater than inside the cell. So if the concentration of the water is outside is greater and inside is less, there will be endosmosis taking place inside the cell. Similarly, exosmosis is the outward diffusion of water through a semi-permeable membrane. So if it is the outward diffusion of water from the cell outside, that is through a semi-permeable membrane, when the surrounding solution is more concentrated. If the surrounding solution is more concentrated, I give you an example of the sugar solution. Sugar solution has got more sugar and less amount of water, whereas the grapes had got more amount of water and less amount of sugar. Okay, so what happens? The water from the grapes are going outside. So when the surrounding solution is more concentrated, this tends to cause shrinkage of the cell. The cell is going to shrink because the cell is going to lose its water. When the cell loses its water, it will become flaccid. It will become losing its water. So it will shrink. So shrinkage of the cell is going to take place when the water is lost from the cell and goes outside into the outer solution. So if the concentration of the surrounding solution is same as the inside of the cell, in which direction will there be? So there will be no movement. So they ask you this question that can you answer this thing, that what will happen if the concentration of the cell and the concentration of the outside is the same. If they are the same, then where will the water go? Whether it will go from there will be endosmosis or exosmosis. So the answer is no osmosis. There won't be any kind of a movement taking place because water will not be able to move anyways because the concentration is same on both sides. So there will be no net movement of the water in such condition as the concentration of water is same on both sides of the cell wall. So now we see a particular experiment about osmosis. It's a very important experiment and we normally follow this. We have normally questions which are asked based on this particular experiment. So let's understand this experiment. So in this experiment, what you do is you take a beaker and then you take a inverted thistle funnel. So you bring a thistle funnel which is inverted like this, and to the edge of the top of the thistle funnel you are going to tie. Means it will be like अगर मैंने ग्लास लिया तो ग्लास के ऊपर जैसे हम लोग एक पेपर या कुछ रखते हैं और बांधते हैं, वैसा करके we are putting a cellophane paper. So there is going to be a cellophane paper. Cellophane paper is a semi-permeable membrane. So cellophane paper is going to be put over here and this whole thing is dipped inside water. Now to the thistle funnel, what you are having is some sugar solution. So inside the thistle funnel, you are going to place some amount of sugar solution. this is nothing but the black part over here is sugar solution, and this part is water. This is cellophane paper. This is a thistle funnel which is kept inverted. So this is the arrangement you have seen. Now you will see that initially the level is this much, and after some time the level would have rise and become this much. So this will be the increase in level, after some time. So after a few hours, when you cleave this thing, the sugar solution of the thistle funnel would be rise. There will be rise in it, and the level of the water in the thistle funnel in the control will remain. So if I have got another one which will be having only water in both sides, so one side no sugar solution, we have just put water in it, so there will be a control setup. See, all experimental setups, there will always be two kinds of experimental setups. One will be the experimental setup and other is a control setup. Experimental setup is the one in which you are expecting some kind of a change to take place. So that is the one which is going to be the one which you are looking forward that okay, there will be some change will be taking place. And the control setup is you are sure that there is nothing going to change over here because there is nothing which can happen. Like if you put some water over here and water over here, you are definitely sure the water is not going to enter from here to here because कोई होने का chances नहीं है. So that's why it will be just remaining as it is. So the control setup will be as it is, whereas the experimental setup there will be an increase in the water level. The level of the water in the beaker in the experiment will drop slightly while the one in the beaker in the control will remain unchanged. So of course in this particular water level in this beaker, there will be a slightly marginal drop in that thing because of the rise in water level over here, whereas control में कुछ फर्क नहीं पड़ेगा because यहाँ पे जितना पानी अंदर गया उतना यहाँ पे available नीचे जाएगा. But normally this is very thin, so you will not be able to make out any difference in the level as such. If the water in the beaker of the experiment setup is tasted, it is not found sweet. Now, important thing. If you put these mixes together, you feel that the sugar from this one should have entered into the water. So this water would turn a bit sweeter, right? You feel that it should turn sweeter, but no. To your surprise, you will find that this will not turn sweet at all. So what does it indicate? What does the conclusion over here? So we conclude three major things over here. First thing, in the experimental setup, some water of the beaker has passed through the cellophane paper to enter the thistle funnel containing the sugar solution. So that's the first conclusion, that water from here has entered here. That's why there is an increase in level. Very easy. That the cellophane paper allowed the water molecules to enter into the thistle funnel and hence the water went over here. And that also because water over here has got more amount of, okay, this is a higher concentration, this is lower concentration, one part. Second, sugar from the thistle funnel has not passed into the beaker. The cellophane paper did not allow the sugar to pass from the thistle funnel to the beaker. That did not happen, and that is the reason the water never tasted sweet. It did not taste sweet because the sugar never entered from here to here. It never went from there to there because the cellophane paper is a semi-permeable membrane which did not allow the bigger molecules of the sugar to move. It only allowed the water molecules to move from inward into the thistle funnel. And the third, the cellophane paper has acted as a selectively or differentially permeable membrane. It has allowed water molecules to pass through but not the sugar molecules. So this is the main thing, that the water molecule were allowed to pass through the cellophane paper but the sugar molecules were not allowed to pass through the cellophane paper, and hence we say that the cellophane paper acted as a differentially permeable or semi-permeable, or we can say selectively permeable membrane which allowed only selected thing to go out and come in. It did not allow the molecules of the water to come out, but allowed only the water to go inside the thistle funnel. So that was about the experiment, and now there is some modifications. We'll just modify this particular experiment a bit, and let's say that if you slightly modify the experimental setup in this by taking a more concentrated sugar solution in the thistle funnel and very little or less concentrated sugar solution in the beaker, the result would still be the same. So even if you are not taken water over here, but you are taken more concentrated sugar over here and less concentrated sugar solution over here, still the result will be the same. That means it is not going to be dependent on, okay, this is only water, that's why it entered inside. No, it is something which has got a higher concentration of water, which allowed the water to enter inside this thistle funnel. If this would have been even a salt water, it would still be water entering here, salt remaining here. This will not become salty because salt will not be allowed to enter inside. So the level of the solution in the thistle would rise, and this again is because some water of the less concentrated sugar solution in the beaker moved into the more concentrated sugar solution into the thistle funnel by crossing through the cellophane paper. But if we are going to replace the cellophane with a muslin or rubber sheet as a barrier, then what would happen is this way, that if in the setup we had used a rubber sheet and in another similar setup a muslin cloth instead of cellophane, we would have seen that there won't be no change in the sugar solution occurring in the first case, that is rubber. So if this would have been a rubber sheet over here, then there would not be any change at all. Why there would be any change at all? Because rubber is a non-permeable substance. It is not allowing the water to enter into this particular area. So in case of I have taken a particular experimental setup in which instead of cellophane paper, if I take rubber, then that level will remain as it is. This level will remain as it is. It will be no change taking place because rubber will act as a non-permeable layer. So the rubber sheet is impermeable and would not allow the water molecule from the beaker to cross over to the other side. So it will not allow the water to cross over. In the second case, the mesh or pores of the muslin cloth are so large that they would not hold back even the sugar molecules, and the sugar solution would flow down to the common level due to gravity. And the muslin cloth is therefore freely permeable for sugar solution. So in case I take the another option of a muslin cloth, if I take a muslin cloth, now muslin cloth is such that it has got too many pores in it, and that pores are so big that easily allows the sugar molecules to pass through them. So because of that, it becomes a freely permeable membrane, and it is a freely permeable membrane. So water will not only, but even the sugars, the water will go here also, and it will be, so it will be something like diffusion taking place, and the whole water will become a sugar solution. So this water will taste sweet if this is going to be a muslin cloth which is over there. So the same experiment can be performed by using a whisking bag. Because it is a semi-permeable. A whisking bag is nothing but a water bag which is having made up of leather kind of material, which will be porous and allows water to flow through it. So that's why the whisking bag, the same experiment, instead of taking a thistle funnel, you can always take a whisking bag and a straw on top of it, and it will give you the same results. So place sugar solution in a knotted whisking bag and insert the long glass capillary tube till some of the sugar solution rises into the capillary tube. Tie the mouth of the bags firmly around the capillary tube and support it with a clamp. Immerse the whisking bag in the beaker with water for about an hour. The level of the sugar in the capillary rises. This rise is due to the water molecule diffusing from the walls of the whisking bag. So here we understand that whisking bag is also a semi-permeable membrane. So here we see cellophane paper and whisking bag are semi-permeable membranes, whereas rubber sheet was impermeable membrane and muslin cloth was freely permeable membrane. So the experimentation of osmosis is going to be holding good only if it is semi-permeable membrane. So in case of a semi-permeable membrane, the water molecules are entering inside, the sugar solution is not going outside, so this water will not turn sweet and the level of the water will rise over here. So that is what is actually going to happen when you are taking any experimentation for the osmosis. So what are the key points? So the key points over here are, there are two liquids of different concentration. So here they are given you this particular figure. So what they have done is they are given you this particular figure wherein you have got this kind of a barrier in between. So this is a barrier over here, and this barrier has got some pores between. These are the empty pores over here, and they have got on one side is a more dilute solution, so it is much more water. So there are molecules over here are less, whereas here the molecules of sugar are much more. So more sugar, this is less sugar. Water molecules more over here, less over here. So water is going to enter from here to here. This is nothing but osmosis, showing the osmosis. Water moving from, so this is higher concentration, this is lower concentration of water. So water moving from higher to lower. So there are two liquids of different concentration. The sugar solution has a higher concentration of sugar molecules. Sugar solution, higher concentration of sugar molecules, whereas there may be fewer or no molecules on the beaker side. So the beaker side is, this is the beaker side having almost no sugar or some very small amount of molecules of that. It is very important to understand that the concentration can also be visualized from the side of the solvent. So you can always see from the side of the solvent also that okay, here more concentration of solvent, less concentration of solvent, or solute wise less concentration of solute, more concentration of solute. You can see it anyways, but mainly is the water is going to go from higher concentration of solvent to lower concentration of solvent. So that is, there would be more water molecules in the unit volume of in the beaker than in the same volume of sugar solution in the thistle funnel. Thus there are two regions of different concentration which may be expressed by the following. The two regions of different concentration of molecules, water molecules, or two regions of different concentration of sugar molecules. So either we can say that this one has got more concentration of sugar, or and this has lower concentration of sugar, or you consider this has more concentration of water and this has less concentration of water. The second thing is, the two liquids are separated by cellophane paper which behaves like a semi-permeable membrane. So this blocks over here are nothing but the cellophane paper, which are semi-permeable membrane. A semi-permeable membrane is one which allows the passage of molecules selectively. It will allow only certain molecules to pass through them and certain molecules to not pass through them. So the one which are semi-permeable are also called as differentially permeable or selectively permeable. It allows the solvent to pass through it freely but prevents the passage of the solute. So that's why these molecules cannot pass through over here, but the smaller molecules of the water can pass through this. Hence it is called as semi-permeable membrane. And then how long will this take place? Till how long will this particular thing happen? Till the time the concentration of this solution becomes equal to the concentration of this solution. Till the time यहाँ से पानी इतना चला गया कि this concentration over here and the concentration over here is equal, will be the condition wherein both the sides of the semi-permeable membrane has got equal concentration of the solution. If both have got the equal solution concentration, then they will be not having any kind of osmosis taking between them. That is called as isotonic, that is same tonicity or same kind of concentration of the solution. So when the solution becomes isotonic, that means they have got equal concentration on both sides of the semi-permeable membrane, then osmosis stops. So theoretically, osmosis should continue until the concentration of water molecule becomes equal, isotonic on both sides of the membrane. In the experiment, the column of sugar solution in the thistle funnel keeps rising upwards with the influx of water from the beaker, and within, the height of the column of the solution would also increase. Such an increase in the height of the rising column reduces further osmosis. A stage will reach when no osmosis occurs even if the concentration of the water molecule is not same in both sides. In this state, the equilibrium of the water molecule from the beaker tends to force upwards through the membrane, but the weight of the pressure from above holds them downwards. Now this is something, it's only experimental over here. Experimentally, what is going to happen here is, when the thistle funnel is over here inverted like this, if the water intake, what is completely keeping on entering into the thistle funnel, what is going to happen is, the level which was here has become now this much. So this much amount of additional water is applying a force on this particular thing. So there is a pressure with which the water wants to go outside the thistle funnel, because the pressure over here is more than what is there inside this particular beaker. So in such a case, when even if the concentration over here is much less than the concentration over here, means here the water level is much more, concentration of water is much more, higher concentration, and here it is a lower concentration, but because of the gravitational pressure, the pressure which is generated by the amount of water which is entered into the thistle funnel, it will not allow, the pressure from the top will not allow the water to enter inside the thistle funnel. So this is just experimentally it is going to happen, that because of the pressure which is coming from the top due to the height of the liquid column, that height of the liquid column is exerting the pressure over here, that it is becoming an equilibrium, that the force of pressure coming from here and the osmotic pressure from here is equal, and hence even if the concentration was different, the osmosis stops. Okay, so we see over here that a stage will be reached when no osmosis occurs even if the concentration of water molecule is not the same on two sides of the membrane. So you can see a stage will be reached when even if the concentration is not the same, the osmosis stops. Why is that so? In this stage of equilibrium, the water molecule from the beaker tend to force upwards through the membrane, but the weight of the pressure is holding it from entering inside. So water wants to enter inside, but this is not allowing to enter inside. So such a condition will be occurring in the end of the experiment, wherein the osmosis would stop because of this thing. Now, so in the above experiment, you can see that if I put a piston over here, if I put a piston, and I take a piston like this and put over here, you will find that the piston is going, and put a weight, it will find that it is going to be pushed upwards. So if air piston bearing some weight was introduced into the thistle funnel from the very beginning, the level of the solution would not rise at all, showing that there would be no entry of water. Thus we define osmotic pressure as, osmotic pressure is the minimum pressure that must be exerted to prevent the passage of the pure solvent into the solution when the two are separated by semi-permeable membrane, or osmotic pressure of a solution is measure of its tendency to take in water by osmosis. So here you see that if the pressure is applied, then it is not allowing the water to go inside. As in the case of the earlier case also, when the water level went so high that it was acting a pressure. So you can see that if the pressure is exerted over here, then that pressure does not allow the osmosis to take place. So when is the osmosis taking place? When the pressure over here is greater than whatever is the pressure over here. If the pressure over here is greater, that means osmotic pressure is the pressure with which there is a tendency of the water to enter into the solution. So osmotic pressure of a solution is the measure of its tendency to take in water by osmosis. So in other words, osmotic pressure is equal to the weight or pressure required to nullify the osmosis. Okay, so osmosis को block करने के लिए, nullify करने के लिए, whatever is the amount of pressure required, that is nothing but the osmotic pressure of this thing. Okay, so that was osmotic pressure. Okay, we will stop here for this video. We'll continue with tonicity the next time.